Ever since hopanoids were discovered in 2.7 billion-year-old shales from Pilbara, Australia, scientists have been researching them to unravel the mysteries of these molecular fossils. Hopanoids, examples of which are pictured here, are pentacyclic compounds that exist in the cell membranes of bacteria. However, their function remains unknown. Hopanoids are the bacteria analogs to cholesterols, which are found in eukaryotic cells. This is due to their similar structure and location in cell membranes. Cholesterol's primary function is to improve plasma membrane fluidity. The molecular pathway to produce hopanoids was probably present in the last common ancestor of bacteria, but was lost by later cells. By discovering the exact function of hopanoids and the resulting structure-function relationship, we will better understand the earliest life on Earth. And understanding early life could even give us clues to understanding the conditions of early Earth.
Our main goal in this project was to explore the role that hopanoids play in methylobacterium. Do methylobacterium with one set of hopanoids function differently? Do they have different phenotypes? Do they grow differently? Much like Goku, our hero from Dragon Ball Z, is challenged by this villain, Frieza, so too are the strains of methylobacteria challenged by his stress. In our experiments, the villain is the stress that is applied to the bacteria's environment. The bacteria die in the face of these stresses. However, different strains of bacteria, which make different kinds and amounts of hopanoids, might be able to fight back better and grow more than others, even in the presence of the stress. Our experiments looked at which strains did better, and tried to detect if hopanoid production could be a reason for this. To take this one step further, some bacteria even evolved to become bigger, better, and stronger in order to fight back against the stress. By selecting for the bacteria that were able to grow the best and fight the stress, we were able to perform evolution in action.
The strain used in our experiments was 2180, which biosynthesizes a unique profile of hopanoid molecules. Our 4 stresses included antibiotics, detergent, and formaldehyde. The bacteria grew on media containing a carbon source called methylamine. We tested the viability of the 2180 strain in the presence of these 4 different stresses and on methylamine. First, we plated the cells on petri dishes. In order to make sure that the bacteria were evenly spread, we added the strain directly to soft agar, which we then deposited onto the plates. To create a concentration gradient of stress from the center of the plate outward, we applied a membranous disc to the plated bacteria. Then, we applied the stress to this disc. After the stress diffused, a circular region of no growth, called the zone of inhibition, was created. The radius of this zone is easily measured and represents the concentration of stress at which the bacteria are viable. The closer the cells grow to the center, the better they are able to tolerate the stress. In addition to measuring each strain's ability to grow in the presence of stress, we also wanted to improve their fitness for individual stresses. To do so, we set up a directed evolution experiment. Using square plates, we set up a similar concentration gradient as in our stress tests, but this time used only formaldehyde. Through genetic variation from mutations, a small number of bacteria are able to grow better than most in the presence of formaldehyde. By picking only these select colonies and transferring them on, we were able to evolve strains that demonstrated greater tolerance of formaldehyde. The goal of this experiment was to reobtain the phenotypic response to formaldehyde that exists in the wild-type strain, which is potentially due to the presence of hopanoids.
Through evolution in action, we are exploring the different roles that hopanoids play in methylobacteria. The use of these stresses can eventually provide insight into their particular mechanism and functions. Given that hopanoids are among the earliest molecules synthesized by life, their currently unknown functions are imperative in understanding the origins of life on Earth.